Hello everyone, and thank you for tuning in today. I'll be presenting my research titled Probiotic Modulation of the Gut-Brain Axis and its effects on recognition memory and hippocampal BDNF in mice. Over the past decade, the gut-brain axis has emerged as a fascinating area of research in behavioral neuroscience. It refers to the complex two-way communication between the gastrointestinal system and the brain. This occurs through neural, hormonal, immune, and metabolic signaling. One of the central players in this system is the gut microbiota, the trillions of microorganisms that live in our intestines. These microbiomes are not just passive bystanders. They are actively involved in regulating brain development, emotional processing, and even cognitive function. One of the landmark studies in this area was conducted by Pseudo and colleagues in 2004. They found that germ-free mice raised in completely sterile environments without any gut microbiomes, had exaggerated stress responses and reduced levels of brain-deprived neurotrophic factor, or BDNF, in the hippocampus. This suggests that the absence of microbiota directly compromises both stress regulation and neuroplasticity. Since then, researchers have started looking at specific microbiotic strains, specifically Lactobacillus rhamnosus, Bifidobacterium infantis, and Lactobacillus helveticus, to see if they could restore or enhance brain function. One influential study by Bravo and colleagues in 2011 showed that chronic ingestion of the rhamnosus reduced anxiety-like behavior and altered GABA receptor expression in the brain. Interestingly, when the vagus nerve was cut, these effects disappeared, suggesting that the vagus nerve plays a crucial role in the gut-brain connection. Now, when it comes to cognition, several studies, including those by Dristuti and Burkus, have found that probiotics can enhance learning and increase hippocampal BDNF levels. But here's the issue. Many of these studies use stress-heavy tasks like the Morris-Water maze, which can introduce confounds by independently altering both brain function and gut microbiota. So, the goal of my research was to take a different approach, one that used a low-stress, more naturalistic test of memory, the novel object recognition task. This study had three main goals. First, I wanted to see whether a 2-week probiotic supplementation would improve recognition memory in healthy adult mice. Second, I wanted to determine if any improvements in memory would be accompanied by higher levels of BDNF in the hippocampus, given that BDNF is a key molecule involved in learning and plasticity. And third, I wanted to explore whether male and female mice respond differently to the same probiotic treatment. This question was based on growing evidence that sex can influence how the microbiota brain connections function. So our hypotheses were as follows. Probiotic-treated mice would show better recognition memory, as measured by their discrimination index scores, and higher BDNF levels compared to control mice would be present. We also expected female mice might show a stronger response, potentially due to the known influence of estrogen on BDNF expression. For this experiment, I used 20 healthy adult C57BL6 mice, 10 males and 10 females. They were randomly assigned to one of two groups, a probiotic and a control group. Mice in the probiotic group received a daily oral garbage for 14 days. The probiotic mixture included 3 strains of probiotics as follows. Each was administered as a concentration of 1 billion colony-forming units per milliliter, and the total volume given each day was 20 microliters. The group received the same volume of vehicle solution without any The control group received the same volume of vehicle solution without any probiotics. On day 15, 24 hours after the final dose, mice underwent the novel object recognition task. This test has 2 phases. First, in the familiarization phase, each mouse explored an arena containing 2 identical objects for 10 minutes. Then, after a 1-hour break in their home cage, they were returned for a 5-minute test phase. This time, one of the objects had been replaced with a new one. The amount of time spent exploring the novel versus the familiar object was then recorded. From this, I calculated a discrimination index using the following formula. This index can range from -1 to 1, with higher values indicating a stronger preference for the novel object, essentially a better memory performance. Immediately after the test, all mice were euthanized under ethical protocols. Their brains were extracted and the hippocampus was dissected and stored at -30 degrees Celsius for later analysis. BDNF protein levels were quantified using an ELISA, or enzyme-linked immunosorbents assay. Each sample was homogenized, centrifuged, and run in duplicate wells for accuracy. For data analysis, I ran an independent samples t-test to compare memory and BDNF outcomes between groups. I also used a two-way ANOVA to check for interaction between sex and treatment. So, what do I expect to find? First, I anticipate that mice in the probiotic group will show significantly better memory performance. Specifically, I expect the discrimination index scores to average around 0.65 compared to 0.45 for the control group, based on research. And that's a fairly substantial difference, and it would suggest a clear preference for the novel object, indicating intact memory. Second, I expect to see a measurable increase in hippocampal BDNF levels. In the probiotic group, around 30 picograms per milligram of tissue compared to just 20 in the control group. Finally, I predict we may see stronger effects in female mice. This idea is supported by previous research from Clark and others showing that gut-brain interactions are often sex-dependent. Estrogen, in particular, is known to enhance BDNF expression, which could amplify the effects of probiotics in females. If our predictions are confirmed, this study would provide strong evidence that probiotics can enhance cognitive function, even in healthy animals, by modulating the gut-brain axis. This suggests their potential is not just as a treatment for clinical conditions, but also as a tool to optimize brain health more broadly. Using the novel object recognition task was a key design choice. Unlike stress-heavy paradigms like the water maze, NOR allows us to assess memory in a natural low-stress context, avoiding confound effects from stress-related changes in the brain or microbiota. The expected increase in hippocampal BDNF provides a compelling mechanism. BDNF is essential for learning and memory. It supports synaptic plasticity and neural survival and long-term potentiation. Finding a behavioral benefit alongside elevated BDNF would suggest that probiotics truly enhance neuroplasticity. And importantly, by including both male and female mice, this study addresses a long-standing gender gaps in neuroscience. If sex differences do emerge, they could inform more tailored probiotic interventions. Estrogen's known influence on BDNF could mean females benefit more from these treatments. These findings also point toward the potential use of peripheral biomarkers like BDNF to monitor probiotic effects in humans, which would enhance translational value. Future studies should sequence the microbiome to identify key bacterial strains, explore whether benefits persist after treatment stops, assess dose-response effects, and test whether similar outcomes are seen in aging or clinical populations. As research into psychobiotics continues to grow, we're beginning to see a future where improving cognitive function could be accessible as adjusting the microbiomes in our gut. And this is so fascinating and should definitely be continued on. Thank you for your time.